Enzyme regulation. Humans don't need all the reactions in the body to occur constantly. Enzymes need to be turned on and off depending on the body's needs. The activity of enzymes is controlled by inhibitor and activator molecules. These molecules bond to the active site of an enzyme or to a different part of the enzyme called an allosteric site and work to stop, inhibit, or turn on the enzyme's function. The quaternary structure of the enzyme plays an important role in substrate and allosteric bonding. Enzyme inhibitors. There are two types of inhibitors, reversible and irreversible. Reversible inhibitors connect with enzymes via hydrogen bonds and hydrophobic connections. After they perform their function, they can release, returning the enzyme to its normal state. They are in equilibrium with their target enzyme, constantly bonding, unbonding, and bonding again. Competitive means that the inhibitor bonds to the active site and blocks the substrate from bonding. Non-competitive is when the inhibitor bonds to an allosteric site, allowing the substrate to bond to the active site, but decreases the rate of the reaction. So in a competitive reversible inhibitor, you can think of that inhibitor competing for the active site in place of the substrate versus bonding somewhere else on the enzyme to then decrease the rate of the reaction. All right, here's a closer look at competitive inhibitors. Competitive inhibitors are similar in shape to substrates. They're able to block the substrate from binding to the active site by binding to the active site themselves. So you can see here, this competitive inhibitor competes for the active site itself, binds to it, and then the substrate is not able to bind. A beta blocker, a type of drug used to manage high blood pressure, is a competitive inhibitor. Beta blockers prevent adrenaline, a protein hormone, from binding to its receptor on heart cells, decreasing the heart rate. Beta blockers occupy the active site of the receptor enzyme. So here's the beta blocker, here's the beta receptor, and it blocks the adrenaline, which is the substrate, from bonding. You see how the green adrenaline and the blue beta blocker are similar in shape, and so the beta blocker also fits part of the active site here, and so it's blocking the binding of this adrenaline. Non-competitive inhibitors. Non-competitive inhibitors bind to a separate part of the enzyme, which you can see here, so not directly to the active site, but in doing so, they cause the enzyme to change shape. This part of the enzyme is called an allosteric site. When the enzyme changes shape, the substrate is no longer able to bind to the active site because the active site also changes shape. The two types of reversible inhibitors act differently on the reaction rate depending on substrate concentration. Competitive inhibitors. For small amounts of substrate, the reaction rate is decreased due to the inhibitor molecules bonding with the active sites. As the concentration of the substrate is increased for a given amount of inhibitor molecules, it occupies more of the active sites and the reaction rate increases. The substrates are more likely to bind to the active site before an inhibitor does. So again, remember that competitive inhibitors act to block the active site, so the substrate can't bond to it. If you have a lot more substrate than you have inhibitor, then more substrate will be able to bind, and that will speed up the reaction rate. But if you have much less substrate than you do your inhibitor, then the reaction that happens at that active site will be more inhibited. For non-competitive inhibitors, no matter how much the substrate concentration is increased, the reaction rate will never reach its maximum value. The inhibitor allows the substrate to bond to the active site, but it has stopped the enzyme from doing its job. Remember, we talked about how that change in shape due to that bonding at that allosteric site changes the shape of the active site and prevents the reaction from occurring. All right, so here is a graph that shows the rate of reaction versus substrate concentration for competitive and non-competitive inhibitors. In purple is just normal enzyme function. Okay, that the rate of reaction will increase up to some maximum value even as we continue to increase the substrate concentration. For competitive inhibitors, remember we're competing for the active site. So as the substrate concentration increases, so does the rate of reaction up to the maximum that the enzyme can handle, even at normal function. But for non-competitive inhibitors, look what happens. We do increase a little bit as we have some substrate, but then we reach a maximum that's much below the production of normal enzyme function, even as we continue to increase substrate concentration. Irreversible inhibitors. Irreversible inhibitors form permanent covalent bonds with an enzyme, changing its shape and inactivating it. It cannot be reversed easily. It is sometimes known as enzyme poison. Even the addition of excess substrate cannot reverse the inhibition. This type of inhibitor is found in natural poisons used by organisms for defense and manufactured pesticides. They permanently bond with their protein target, destroying its functionality. Artificial inhibitors are used in medicine for destroying malfunctioning cells in organism. For example, chemotherapy for the treatment of cancer. Ribosome inhibiting proteins, or RIPs, are irreversible inhibitors. Ricin, a protein found in castor beans, irreversibly binds to glycoproteins. When ricin binds, the ribosomes change shape and cannot synthesize new proteins, which kills the cell. Enzyme activators. These molecules bind to enzymes and increase their activity. They bond to an allosteric site, leaving the active site for the substrate. Cofactors and coenzymes, or enzymes that bond to an active site, helping another enzyme catalyzing a reaction, are enzyme activators. Feedback loops. A feedback loop is the path that leads from the initial generation of a signal to the modification of an event. Feedback loops can be positive or negative. A positive feedback loop is where a signal is continually amplified until a desired outcome is reached. The key to these loops is that any small change will be amplified. A negative feedback loop occurs when the outcome of an action reverses the cause of the original signal. Let's take a closer look at a positive feedback loop. As the amount of product increases, it sends a signal back to enzyme 2 to increase the reaction rate, thereby increasing the amount of product produced. The rate is increased exponentially and needs to be turned off at some point by negative feedback. A positive feedback loop works a little bit like a snowball rolling down a hill. Rolling down the hill you can think of as continuing to produce more and more and more and more product at an exponential rate as that feedback is occurring, but then eventually when we get to the bottom of the hill, we have to slow down. So something eventually has to work in opposite of that positive feedback loop. Here are some positive feedback examples. As contractions happen during labor, the hormone oxytocin is released into the bloodstream. As oxytocin levels increase, more contractions occur until the baby is born, which stops the feedback loop. Another example involves lactation. The more a newborn baby suckles, the more milk is produced. This is due to a positive feedback loop involving the hormone prolactin. Positive feedback modulation. For positive feedback, an off switch is required. If muscle contractions are a positive regulator for oxytocin production, then there has to be some way to turn that off. For nursing, the letdown response and baby latching on will begin to turn it off. During labor and childbirth, transition will turn it off. Specifically, oxytocin secretion is suppressed by relaxin, an ovarian peptide that suppresses uterine contractions and relaxes pelvic connective tissue during childbirth. Relaxin acts in a negative feedback loop, to be explained next. Negative feedback loop. So, as the amount of product increases, it sends a signal back to enzyme 1 in this case. And that signal says, reduce the reaction rate, reduce the reaction rate. And what that will do is slow down this reaction and thereby reduce the amount of product produced. This is also called feedback inhibition. When a thermostat senses it is too hot, it turns on the air conditioner to cool it off. If the house is too cool, it will send a signal to turn off the air conditioner. That's that negative feedback loop. We have to have an off switch, some way to turn off that increase in production. Here are some examples of negative feedback. Most control systems in the body involve some sort of negative feedback system. Cells send signals to other cells to fix problems they're sensing. This could involve releasing another signal to counteract a problem, or it could involve the shutdown of the original signal. Examples include body temperature control, the regulation of pituitary hormones, and the control of blood glucose levels. Let's look closer at pituitary hormone regulation. Negative feedback loops can dampen a response or can modulate a metabolic pathway that maintains a steady state level of a product. They decrease the synthesis of a product when enough has been made. They are often off switches. Negative feedback loops play an important role in maintaining homeostasis. Glucocorticoids are synthesized in response to stress, for example, during the flight or fight response. When glucocorticoids increase in a person's bloodstream, they have many potent systemic effects. Prolonged production would be a problem. In addition to increasing in the bloodstream, receptors in the hypothalamus and pituitary also receive glucocorticoid signals. The hypothalamus responds by stopping production of CRH, and the pituitary stops making ACTH. Once glucocorticoids have had the desired effect, the production stops. While glucocorticoid control is an example of dampening a potent response, too much glucocorticoids in the bloodstream has to be negated by the negative feedback in the pituitary and the hypothalamus. The synthesis of ATP is an example of a pathway that is usually under steady state control. Cellular respiration is comprised of a catabolic pathway made up of dozens of enzymes. ATP is the product, And ATP also functions as an allosteric inhibitor of some enzymes in the pathway. When ATP in the cell is low, there is little inhibition of enzymes by ATP, and catabolism proceeds to make more ATP. When there is a lot of ATP in the cell, ATP inhibits enzymes in the pathway, and production of ATP slows down. So for ATP, we need to try to maintain this steady state, this steady level. So we have this system on either end to either increase the production or decrease the production of ATP. ATP is produced at 3 points in cellular respiration. Here, here, and here. ATP is an allosteric inhibitor of phosphofructokinase, the second enzyme in the pathway right here. ATP slows down its own synthesis so that ATP is maintained at steady levels in the cell. So as ATP is produced here, we have this inhibition of the production of phosphofructokinase to maintain steady levels in the cell. Here's another example of a negative feedback loop. Hyperthyroidism is a condition in which the thyroid gland makes too much thyroid hormone. The condition is often referred to as an overactive thyroid. This comes from the U.S. Department of Health and Human Services. This disease, hyperthyroidism, affects more than 5% of women in the United States, 10 times the rate we see in men. One sign of hyperthyroidism is swelling of the thyroid glands. The thyroid gland controls how much energy is being produced by the body by increasing or decreasing the amount of a thyroid hormone, triiodothyronine, or T3, that is circulating in the blood. The thyroid is controlled by the brain, which monitors levels of thyroid hormone and adjusts its signal to the thyroid accordingly. So the brain signals the thyroid to produce these thyroid hormones. This is known as a feedback loop. Thyroid stimulating hormone, or TSH, acts as a coenzyme in thyroid cells that then activates the metabolic pathway for the production of T3. So increased TSH leads to more T3, and increased T3 production means more energy. When the brain has determined that there is sufficient T3 in the blood, it slows the release of TSH, so the thyroid reduces the production of T3. So the brain is monitoring this production of T3 and adjusts the release of TSH accordingly. In this way, the brain is exhibiting allosteric regulation of the enzymes in the thyroid via release of a coenzyme, TSH. Allosteric control. The enzyme at the start of the metabolic pathway that produces T3 requires TSH so it can function. So here's the enzyme 1. TSH stabilizes the active site of the enzyme, allowing it to bind with the substrate. The metabolic pathway that creates T3 will now be discussed. So here's TSH, here's our enzyme, and here's our active site that only works when TSH is present. So we're talking about this point in our process. This is our enzyme 1, and this is the active site for that enzyme that only works when TSH is present. Without TSH, substrate B, the next thing we produce in this process, cannot be produced and the pathway is shut down. The active site deforms without TSH and the substrate is unable to bind and no T3 is produced. There are millions of enzymes. So the amount of TSH available is what dictates how much T3 the pathway will produce. In the example on the left, we have lots and lots and lots of TSH. Excess TSH will produce the most T3 product. Smaller amounts, though, will produce less. If the thyroid regulation is lost, then hyperthyroidism may occur. So if we don't have that regulation where the brain is listening to see, should I produce more TSH to make more T3, or do I have enough T3 so I can slow down my production of TSH? If that's not working properly, we can get hyperthyroidism, and it can happen in many ways. One way is for enzyme 1 to be defective and remain on even if TSH is present. A substance similar to B or C might be present in the system, so the regulation of enzyme 1 does nothing. Another way is that the brain can fail to recognize too much T3 is present and just continue making TSH regardless. Or the TSH molecule is defective and bonds too strongly to the enzyme, causing increased activity. In other words, the TSH does not break down normally.